Hello, everyone. Welcome back to the second half of Biology 100. As many of you probably can imagine, this is not how we all thought it was going to go down, but for the rest of the semester, we will be online. So I will be giving you lectures every Tuesday via this online format called Zoom. Basically allows me to record your lectures over the PowerPoint and then post them either on Zoom or Blackboard. I'm going to be doing this lecture in two parts. So about the first half will be part one, and then the second half, the last roughly 29, 30 slides, will be the second half. This is my first time doing online recorded lectures. So if there's just general feedback you have, like if you want me to talk slower, if you need me to point more things out on screen, if the audio is not good enough, if it needs to be louder, whatever feedback you have will be greatly appreciated. But in terms of jumping back into the material, this week we are on chapter 10, which is all about evolution and natural selection. So we will go ahead and get started. So objectives for this chapter. We're going to talk all about evolution in action. So what does it mean? How does it apply to various organisms? What are a lot of examples of it? Where did it come from? All of that good stuff. We're going to talk about Darwin, Charles Darwin. He's the founding father of the theory of evolution, and talk about his history and the different theories that gave rise to his ideas, and ultimately how he came up with the theory of evolution himself. We're going to talk about the four evolutionary mechanisms that help us explain why evolution occurs. Additionally, we'll talk about why populations are able to adapt in their environment through natural selection based on certain traits they have that allow them to evolve overall. And lastly, we're going to talk about the occurrence of evolution. So what evidence do we have to actually prove that this theory is sound? So the chapter starts off with this example of evolution with fruit flies. What they do, they show us this fruit fly population of Drosophila melanogaster, and they basically do this starvation experiment where they have all of these flies. If you look at this graph down here, the number of flies are on the y-axis, then the hours till death from starvation are on the x-axis. So we have this general curve, and with the mean, the average amount of time until starvation or death from starvation in the middle, about 20 hours. So we have this roughly symmetrical curve of a distribution of the population. And what they do is they perform this starvation experiment. They basically take a population of the fruit flies, which are all of the organisms that occupy the same space in the same region. In this case, they're all flies that inhabit this testing environment. And they basically take all of the flies they have, and they take the food out, and they see who lives and who dies. And all of the ones, after a set amount of time, that survive the starvation experiment, they breed, and they produce a new generation of fruit fly. Those fruit flies, who are then the offspring of the initial survivors, then go through the starvation experiment again. For a set amount of time, they're deprived of food. The survivors from those experiments then breed, and then their offspring are then put through the starvation experiment again. The survivors from that experiment then breed, and they do this over and over and over and over again. So what we see when they do this is that if you look on this box on the right-hand side, it says evolution in action. We have a graph for generation 0, that's the initial population, and we have the number of flies on the y-axis and the hours until death from starvation on the x-axis. So our initial population on the previous slide we saw was 20 hours. That's the same graph we see up here. We just see a larger scale going to 200 hours. So the initial population only survived for 20 hours on average. But as we perform that starvation experiment, the offspring, we notice, start to creep up a little bit. That average is now getting a little closer to 30 for those offspring, for that generation 1. Then from continuing to do the experiment over and over and over and over again, we push this curve from 20 to 30 hours all the way up to an average of 160 hours. So basically, by manipulating these organisms in this environment, we actually were able to drive natural selection, which we'll talk about later in this presentation, and drive the population to have a certain characteristic. That certain characteristic was the ability to be able to survive for longer periods under periods of starvation. So we actually drove evolution through this experiment. And ultimately, evolution is just a genetic change in a population. With the example of the fruit flies, we had the flies that died and the flies that survived from the starvation period. So certain genes allowed the flies, for whatever reason, to go for longer periods without food. And we basically selected for those genes to have the offspring survive longer periods of starvation, thus changing the genes in the population. So at the end of those numerous amounts of experiments, we were able to have flies that could survive very long periods. So evolution is just a genetic change in a population. Doesn't matter what kind of genetic change, just a genetic change. And ultimately, evolution occurs through what's called natural selection. So this is just the idea that certain individuals in a population are born with certain characteristics or traits that allow them to survive better than others. And this will vary from environment to environment. So for example, something like a desert, organisms that are able to maintain water very efficiently when there's not a lot of water around are going to be more likely to survive than organisms that are constantly wasting water and are not as physiologically adapted to surviving that dry temperature. And there's lots of examples we'll go through. But before we dive into all these specific examples, we're going to do a bit of a history lesson. So a good amount of this chapter covers the initial lines of thinking with evolution and how Darwin actually came up with his theory of evolution and how ultimately we see the theory of evolution today. So we're going to sort of dial it back to the 1700s and go through all of the different influences Darwin had. So the very first influence that Darwin had, his name is George Louis Leclerc. Basically, his big contribution to Charles Darwin's influence and way of thinking with the Earth was much older than we actually thought it was. Originally, a lot of people from influences from the Bible thought that the Earth was only a few thousand years old. Geologically, Leclerc was actually able to prove that the Earth was about 75,000 years old is all they were able to determine at this point. And it's not quite as accurate as we have nowadays, but it was a big step in thinking at the time for geologists, as well as many just independent thinkers at the time. So Leclerc is all about proving the Earth was much older than we previously thought from the Bible. The second big influence is Georges Cuvier. And Cuvier was a big contributor through the fossil record. He was able to show that fossils allowed us to see what organisms were still alive and what organisms that were extinct. Because through much of the religious philosophy at the time, it was believed that all of the organisms currently on Earth were there since the beginning. They just were created, and everything that's alive now was automatically alive back then. Cuvier showed through the fossil records that that's not necessarily the case. We do have organisms that are no longer with us that used to be alive thousands and possibly even millions of years ago. So that was his contribution. Third, we have Jean-Baptiste Lamarck. And Lamarck was probably the first thinker that really sort of kickstarted these ideas of actually talking about what evolution is. Maybe not using the same terminology that we use nowadays, but he had an initial idea. And that idea was this idea that species change over time. And what that means is that they have what's called, or he quoted them being called, acquired characteristics. So basically, an organism was able to acquire some sort of trait or skill that allowed it to survive its environment. And this is a very sort of, very rough around the edges way of thinking about evolution. And it didn't quite pan out to be correct, but this was, again, a step forward at the time. Next, we have Charles Lyell. And Lyell was a geologist. So he was very interested in looking at the Earth and all of the features and how they constantly change. And basically, his big contribution to Darwin was showing that the Earth has all of these geological formations that are constantly in flux. The Earth isn't a stable rock formation. We have moving plates, we have mountains and trenches. All of these things are constantly changing. They're just changing over a very long period of time, so we don't notice them as much. but these potential geological shifts actually do contribute to biodiversity. So all of these influences went into Charles Darwin's thinking. So now we're going to actually dive into Darwin's journey. Okay, so with Charles Darwin's history, he was born in the 1800s in England. Ultimately, he decided to go to university at age 16. The school system for the 1800s was a little different than it is nowadays, so people did go to university during their teen years. But he went there for medical studies, and he was not a very great student and was actually found to be not doing well in his classes. He wasn't very interested in the material, and he actually was kind of grossed out by some of the medical stuff that he had to do and learn about. So he wasn't very invested in his field. Ultimately, he decides to drop out, and eventually he shifts majors, if you will, and studies at a new university called University of Cambridge, and he studies theology. So theology is the study of God, so religious studies. Wasn't super crazy about that either, but he had external influences and pressure to go to college and get a degree and move forward with his life. Ultimately, though, he was very in love with nature. He loved looking at the natural world. He loved looking at observations, studying how ecosystems work, how the environment comes into play. All that was his real passion. And ultimately, Darwin in 1831, so after he finishes at the University of Cambridge, basically gets what is referred to by your book as his dream job aboard the HMS Beagle in 1831. What the actual job description was that he was a gentleman companion to the captain. And what that actually meant was that during the voyage to the New World, or the Americas and all around the globe, he basically was a intellectual consult for the captain. The captain was actually the captain of the HMS Beagle was actually very intelligent, but most of the crew workers were uneducated at the time. So he didn't have someone to talk with him on the same caliber of intelligence that he wanted. So the captain basically hired him as an intellectual consultant for the journey. And the journey started all the way from London, England, and they sailed all the way down across the very southern tip of South America, up here to this group of islands called the Galapagos, which we will dive into in a second, around the Pacific Ocean, back along the southern part of Australia, through the Indian Ocean, back through the Cape from South Africa, and then back up to England. So they went around the entire world. Didn't stop in every country, but they made a lot of stops along the way and gathered a lot of key information with species and plant and animal samples for studying nature and evolution. Ultimately, Charles Darwin's big observations were on the Galapagos Islands. So the Galapagos Islands is this little dot here of islands that are in the area just off the coast of Ecuador of South America. So that's where the big revelations that Charles Darwin had were. So Charles Darwin made two very important observations during his journey. He looked a lot at the physical characteristics, which we know as traits of organisms, and ultimately pointed out two things. So the first thing was while he was on the Galapagos Islands. So on the Galapagos, there are bird species known as finches that all seem to resemble the mainland species. You look at things like beak shape, like feather color, and notice that there were similarities between those finches that were on the islands versus those that were on the mainland in Ecuador. And the second big observation he saw was that during his trip, he was able to see a lot of fossils of both extinct species, but also of species that were living in the same area. So he basically was able to make a connection with the fossil record that provided these connections with different kinds of species. One of the big examples that your book gives is something called the glyptodonts. So there are these guys right here that I'm circling. And in Argentina, he was able to show that these organisms, the armadillos that we have nowadays, may have come from the Glyptodons. So he made connections on the Galapagos Islands with the finches, and then he made connections with the fossil record through various parts of the journey he went on. So he gets back to England, and basically over a course of years, he starts to write up his ideas, kind of put everything together. He doesn't have the theory of evolution quite yet. He reads an essay called The Principle of Population by Thomas Malthus. And basically, Thomas Malthus shows that populations are able to grow much faster than food supplies. So basically, give it enough time, there won't be enough food to supply a group of organisms. And ultimately, different kind of factors will sort of control that population. And Charles Darwin's big idea at the time was that his quote is that favorable variations would tend to be preserved and unfavorable ones tend to be destroyed. What that basically means is that for any given population, there are winners and there are losers. The winners are able to survive and produce offspring and carry out their lineage onto a new generation. The losers, quote unquote, are basically, they die. They don't get to produce offspring. So they are the ones that have the unfavorable outcomes, and the ones that have the favorable variations or outcomes live on, they're preserved. So Charles Darwin writes up this draft in 1842. It's a 35-page draft of all of his ideas. And for whatever reason, he shelves it. He puts it in a desk drawer and he doesn't look at it again. We're not really sure why he did this, but basically he doesn't publish it immediately. And it basically stays in that drawer for years and years and years. What happens next is that an individual called Alfred Russel Wallace basically is able to also piece together this idea of evolution by natural selection. And in 1858, he basically publishes a smaller piece outlining these concepts overall. And Darwin is kind of, Darwin is basically upset and a little depressed because someone beat him to the punch. Because he had these ideas and someone sort of got there. But Wallace didn't have quite the whole picture. And many of Darwin's friends basically tell him that you have some ideas and Wallace has some ideas. We'll bring this to the Linnaean Society in London and propose this theory of evolution and ultimately show that you both get credit for it. And that is actually what ends up happening. But Darwin still wants to write about his findings that he found on his voyage. So he basically contacts Alfred Russel Wallace and basically says, hey, I know you've done most of these ideas, but I also have some views on this, but I feel like I'm kind of copying you. What should I do? And Wallace basically tells him, if you feel you have original content, like you should share it with the world. That's your sort of responsibility as a scientist. So Darwin writes up, takes that copy out of the drawer, finishes up his book, and in 1859, he publishes The Origin of Species. And that basically takes some of Wallace's ideas that have already been established, along with what he learned with evolution by natural selection, and it sort of coins the term descent with modification. So what that basically means is that organisms are changing from every generation to new generation. They're being modified. So your descendants are the individuals that come from your parents, and they're being modified by either the environment or some sort of external force. This is a radical new idea for the time. The book that Charles Darwin has sells out very, very quickly. And ultimately, he receives pretty much the bulk of the credit for the theory of evolution. Alfred Russel Wallace does take some credit, but in terms of how the history books remember it, Darwin is really the one who pieced it all together and got all the ideas down on paper. So that's why, like when we had Darwin Day back in February, we call it Darwin Day, not Alfred Russel Wallace Day. So interesting piece of trivia there. Okay, so moving on. Okay, so that is all the history behind Darwin, how he comes up with the actual idea of evolution. So now we're going to move into what all that actually means with evolution. So evolution is all about changing some sort of genetics or genetic characteristics. And another way of putting this is that the allele frequencies in a population change. For example, we have on the left this population of tigers. We have a recessive white tiger and a more dominant orange tiger. So if we look over here on the right, the allele frequencies in the tiger population, we have the orange piece of the pie chart representing the orange tigers, and then the white sliver representing the white tigers. So a vast majority of the population has the phenotype of orange coat, and then a very small amount of the population has the white coat. Over time, we have a shift in the amount of allele expression. So we're now starting to see in the middle graph that a little more of those recessive white alleles are being expressed. And then after a little more time changes, we see a good amount more of that white allele is now being expressed for the whole population. Meaning at the same time that that orange allele is now decreasing. So going from left to right, we now see that less of the pie chart is now orange, more of it's white. So we had a change in the allele frequency. The orange allele went down and the white allele went up. That was evolution. We had an allele frequency change, we had evolution. The important thing to note with evolution is that populations of organisms are the ones that undergo evolution. Individuals, however, do not undergo evolution. So when we talk about evolution, we talk about a population evolving. We don't say an individual tiger evolved, we say a population of tigers evolved, because we have to be able to track the allele frequencies from generation to generation across multiple different organisms and going from offspring or going from parent to offspring. So populations evolve, organisms do not, is the takeaway there. Okay, so now we are moving on to the mechanisms of evolution. So 4 different mechanisms of evolution we have. They are mutation, they are genetic drift, migration, and natural selection. And your book describes these mechanisms occurring when what's referred to as the market share of an allele changes. All that means is the frequency changes. So that means, does the frequency of an allele go up, or does the frequency of an allele go down? If it goes in either direction, doesn't matter if it's up or down, increasing or decreasing. That is evolution. So the 4 ways that this happen are mutation, genetic drift, migration, and natural selection. And we will go through each in turn. So mutation is all about changing the DNA. And we talked a lot about this when we were looking at transcription translation with going from DNA to RNA to proteins. We know that either from in a spontaneous mutation, so one that just happens naturally, DNA can change. We can change one of those DNA base pairs, so your adenine, your thymine, your guanine, or your cytosine. Or we can have it externally occur through something like UV radiation, potentially a virus could change DNA. All of those are external factors. But any kind of base pair change we have to the DNA itself is a mutation. And ultimately, this is a form of evolution because we know that changing the base pairs codes for some sort of gene, which codes for some sort of RNA, which codes for some sort of protein. So there will be a change. For mutations with evolution, we only are really focusing on mutations that affect the reproductive cells and can be passed on to the offspring. For example, an individual may have a mutation on a sex chromosome that leads to Down syndrome, for example, like we talked about in the last unit. That would be a mutation that we're interested in evolution because we're talking about a condition that's affected at the genetic level and can be passed on from passed on from parent to offspring. Something like cancer, for example, that is a mutation that affects the individual, but you are not going to pass on that cancer to the offspring. So we're really looking at mutations that can be passed from parent to offspring. And like I said, mutations can be caused by either environmental factors, so external factors at play, or they can just happen spontaneously, just sort of by accident. Ultimately, though, they are random. So we don't know ahead of time who's going to have a mutation and who's not going to have a mutation. We don't know if, for example, someone is exposed to UV radiation, they may have a bunch of mutations, they may have no mutations. We don't really know what's going to happen because we don't have the ability to track those. Additionally, when a mutation occurs, we aren't necessarily able to predict the outcome of that mutation. So a mutation could potentially be benign, meaning that it doesn't actually do anything, it's just neutral. It could cause a harmful effect, so it could produce a misfolded protein, which doesn't do its job right. It could lead to a genetic disease. It could have some sort of detrimental impact on an individual in a population. Or it could be useful. When talking about evolution, it's possible that a mutation grants an individual a new characteristic to be able to survive in their environment better, and then pass on their genes to the offspring that allow them to survive better. So we ultimately don't know if mutations will be good, bad, or neutral. That's sort of something we can only understand on the back end after it's happened. So with mutations, they are ultimately the ultimate source of genetic variation in a population. Of the 4 different kind of mechanisms we have, mutations are the best source of genetic variation for population. So having either mutations come from an external source or mutations come from a spontaneous mutation, that's what's really going to drive genetic variation in a population. And like I said, mutations may either not impact an individual's fitness level, and we'll talk about fitness in a little bit. They could cause death early through some sort of detriment, or they could potentially reduce an organism's reproductive success. So we don't know specifically what's going to happen with that mutation, or a mutation in particular, when it happens, but we know that there's one of 3 things that could happen: good, bad, or nothing. Okay. The second form, or the second mechanism of we're looking at is genetic drift. And genetic drift is a change in allele frequencies in a population. However, this is occurring randomly, and the idea is that with genetic drift, there is some sort of external force that's occurring that's changing the population in some way. And ultimately, genetic drift can lead to fixation, and that's basically when an allele's frequency becomes 100%. So for example, if we had a dominant allele and a recessive allele, and the dominant allele got to 100% of being expressed, so we have no more recessive allele, that's not very good for a population. For any sort of population, you always want to have a genetic variation. Genetic variation allows a population to survive over the course of generations and be ready to tackle any sort of condition or problem that the environment throws their way, whether that's a natural disaster, whether that's new predators or less predators, whether that's increased competition for food, whether that's a genetic disease or virus. Any of those are potential things that you want to have variation for in a population. And we have 2 specific types of genetic drifts. The first is what's referred to as the founder effect. And the founder effect is when you have a small number of individuals that are going to leave a population and basically become the founding members of a new population. So if we look on this pie chart right here, we have our source population that I'm circling right now. And we have the sort of pukey green dominant allele frequency and the turquoise green recessive allele frequency. So we have mostly dominant allele and not a lot of recessive allele. Ultimately, we're going to have a chunk of this population, the pie wedge that's being broken out, and that's going to move on to a new area and found its own population. So now we have a new population that has a certain portion of the dominant allele, and now a larger portion of the recessive allele. So by founding that new population from individuals in that population leaving and forming their new population, we now have a different amount of a different ratio of dominant allele expression to recessive allele expression. So that's the first way that genetic drift occurs. The second way that genetic drift occurs is called the bottleneck effect. And the bottleneck effect is when you have a population that gets quickly reduced to a small fraction of their original size because of some large external change. So for example, with our source population up here, we have a bunch of different alleles, not necessarily dominant and recessive in this case, but we have blue, purple, yellow, and green. And there are just four alleles we have for some trait. And we can have what's called extreme and rapid environmental change. For example, let's say we had a fire. So we had a bunch of squirrels in a forest, and we had a forest fire, and that burned down a lot of the forest, and a lot of the squirrels died, unfortunately. So what happened through that huge environmental change, that was an external factor, that destroyed a lot of the forest, and that killed a lot of this genetic variation. So we no longer have any yellow or purple. The only survivors that are left are the blue, or the squirrels that have the blue gene, and the squirrels that have a little bit of the green gene. And they ultimately will form that new population of squirrels. We refer to this as a bottleneck effect because you're basically bottlenecking or forcing a large population to then be squeezed into a narrow amount. So in this case, the bottle had 4 colors, and then because of the forest fire in this example, we basically bottlenecked or squeezed out most of the population, and we kept only a few survivors that have a lot less genetic variability than the original population. So that is the bottleneck effect. So that's our second form of genetic drift. All right, hold on 1 second. I need to fix my notes. Okay, okay, so our third mechanism for evolution is migration. So the migration is all about gene flow. And this is when you have the movement of certain individuals of some kind of species. So let's say tigers, for example, and some of them move from one population to another. So either some tigers move from their source population out of their source population, or maybe we have some tigers from outside the source population move into the source population. So we can have individuals move into the population or individuals move out of the population. In your book is this example on the right with gene flow that you'll have 2 populations separated by this mountain range of the same species. And we have a different amount of genetic variation. So for one gene, it's the turquoise, we have a certain amount, and we have a lot more of the pukey green gene. And for population 2, on the other side of the mountain, we have a lot more of the turquoise gene and just a small sliver of the pukey green gene. And ultimately, we can have individuals from population 1 with a larger portion of that pukey green gene move across the mountain into the second population, thus changing their genetic variation because we had gene flow. We had individuals in population 1 carry their genetic information to population 2, and then by being added to that population, we changed the genetic variability of that population. So that's our third form of, that is our third mechanism for evolution, is migration. Our last mechanism for evolution is natural selection. So this is the sort of big famous one that Charles Darwin is known for. So natural selection has three conditions that have to be met for it to occur. Ultimately, natural selection is telling us, is nature basically picking and choosing those winners and losers that I referred to earlier with Charles Darwin reading Thomas Malthus's book. And the winners and losers are selected by the environment. For natural selection to actually occur, we have to have three conditions. So we have to have variation for a trait in the population. We have to have that variation be heritable. And the individuals from one version of the trait have to produce more offspring than individuals in a different version of the trait. What that means is it's called differential reproductive success. And we're going to go through all 3 points piece by piece. So, condition 1 is variation for a trait. What that means is that you have an expression of different alleles. You have some individuals that express the dominant allele, some individuals that are heterozygous and they have the dominant allele and recessive allele, but the recessive allele is masked. And then some individuals have the recessive allele, or both copies of the recessive allele, and are homozygous recessive. So we have to have variability. If we don't have variability, we can't have natural selection. And the example your book gives is with dogs being of so many breeds and varieties, since we have artificially selected them to produce a lot of different traits. So variability is the first condition that needs to be met. The second condition that needs to be met is heritability. We talked about heritability in the last chapter with genetics, but heritability, again, is just the idea that we pass on our genes from our parents to our children, and that those genes are then inherited by those children. So that's why parents look similar to their offspring. That's why certain ailments are passed on from parent to offspring. That's why certain gifts or skills are passed on from parent to offspring. It's all genetic. It's all heritable. So that's our second condition for natural selection to be met. Our last condition for natural selection to be met is called differential reproductive success. So what that means is that not every organism is going to be able to produce the same amount of offspring. Certain organisms are going to be able to produce more, some are going to produce less. But ultimately, we're going to have more organisms that are going to be born that survive. And these organisms that are born are going to struggle for existence. So they are not necessarily going to be winners just by being alive. They have to have some trait that they have that allows them to exist, that allows them to not get weeded out. And since some organisms are more likely to survive and reproduce, they will pass on their genes to the next generation. So it's called differential reproductive success because not everyone has equal opportunity to reproductive success. Some people are going to have greater opportunities. Some people are going to have less great opportunities, depending on the environment and what genes are currently in play for that population. So I'll give a brief recap of natural selection. With natural selection, the 3 conditions that have to be met are that we have to have 1, the variation for a trait. So we have to have different versions of whatever the trait we're looking at is in a population. It can't just be everyone has the same trait. Otherwise, there's no variation. 2, those traits have to be passed on from parent to offspring. And then 3, we have to have differential reproductive success. So we have to have certain individuals that have a trait that are granted more reproductive success, and we have to have individuals with other traits that may not be granted as much reproductive success. All 3 of those conditions have to be met for natural selection to occur. And again, natural selection is basically the environment choosing what organisms, based on whatever genes they have, are better suited for the environment or not as well suited for the environment. And the ones that are better suited for the environment are going to reproduce and survive. the ones that are less suited for the environment are not going to make it. They're going to die and not pass on their genes because they don't have any offspring. And natural selection, by way of the process of those three conditions, is able to eliminate traits that confer poor reproductive success. So the example that your book gives on the right is that we have running speeds of rabbits vary from one individual to another. Some rabbits may be very quick. They're able to run really quickly in a short amount of time. Some may be average, and some may be not so great. Ultimately, we have a variation for a trait, running speed. Not every rabbit can run at the same rate. Additionally, those traits are able to be passed on from the parent to the offspring. So our yellow slow rabbits will pass on their lack of running speed to their offspring, so they won't run as quickly. The medium pukey green rabbits will pass on their genes and have medium running speed rabbits. And then the turquoise rabbits will pass on their genes and have very quick running rabbits. And then with our third condition, our differential reproductive success, we have a population of rabbits whose natural predator is the fox. And the fox is going to eat the rabbits, of course, but most likely the foxes are going to be able to eat the slower rabbits very easily because they're not going to run very quickly and they won't be able to get away. And the fox won't have to expend as much energy to get a meal. If the rabbit's moving slow, why waste energy? You don't have to. Why would you chase the faster rabbit if you didn't need to? So they're going to go after the slower and the medium rabbits and kind of start to weed them off. So ultimately, those faster rabbits are going to have more survivors. They're going to be able to have more babies, and they're going to be pass on more of their faster running genes to subsequent populations. So the faster rabbits have a higher reproductive success than the medium and the slow rabbits. And the last slide for natural selection related to this is a special type of selection called sexual selection. So sexual selection is sort of a offshoot of natural selection. And this is the idea that certain traits for individuals are more attractive to the opposite sex. For example, peacocks have brightly colored feathers that can be used for display from females to males. And for example, large antlers on male red deer are a big trait of sexual selection for male deer showing off to female deer. Basically, the idea behind sexual selection is that there's some sort of ornamentation or display that an individual can do to a member of the opposite sex and basically show them that they have the ability to have greater reproductive success. And by that, usually females, who usually provide the choice for a population, they will choose males that have characteristics that they find desirable, and they will breed with them and produce offspring. So it's not necessarily the environment is not selecting who lives and who dies in this case. What's happening is the females are selecting who they're going to mate with, and ultimately that's going to dictate what genes are passed on. So ultimately, sexual selection, sort of by proxy of natural selection, can change allele frequency based on how females are choosing their mates. Okay, so those are the main mechanisms for evolution. Now we're going to move on to talking about allele frequency with the Hardy-Weinberg law. So we will be covering this very extensively next week with lab. The entire lab, I believe, is dedicated to Hardy-Weinberg, but we're going to go over the basic features of what the Hardy-Weinberg law is, as well as what does it all mean and how does this apply to evolution and expression of alleles. So the Hardy-Weinberg law is basically just a mathematical formula that helps us determine the frequencies for your dominant alleles and the frequency for your recessive alleles. The formula is very, very simple, and we can put it into practice to determine the frequency for alleles. So the formula overall is P plus Q equals 1. And what that P stands for is the frequency of the dominant allele. So P by itself is dominant allele frequency. And then we have Q, and Q is the frequency of the recessive allele. So ultimately, when we're talking about frequency, frequency is always going to be between 0 and 1 for both the recessive allele and the dominant allele. That frequency is basically the percentage of the population that has that allele. And since we're talking about frequency, when we add up all the frequencies together, the frequency of the dominant allele plus the frequency of the recessive allele should always equal 1. Whenever we talk about frequency, they always have to add up to 1. So we will take this one step further and apply this to our homozygous dominant, our homozygous recessive, and our heterozygous dominant. So we know that our P represents the frequency of our dominant allele, and the Q represents the frequency of our recessive allele. So for our dominant allele, to figure out the frequency of our homozygous dominant genotype, so they have 2 dominant alleles or 2 Ps, we are just going to take that P and we are going to square it. So P times P is the frequency of a homozygous dominant genotype. For our homozygous recessive, we have 2 copies of the recessive allele. So 2 lowercase letters of whatever gene we're looking at, lowercase a, lowercase l, whatever gene we're talking about. So for Q, we're going to take 1 recessive allele and multiply it by the 2nd recessive allele, and then I'll get the frequency of our homozygous recessive genotype. Lastly, for our intermediate, for our heterozygous dominant condition, we are going to multiply the dominant allele, Rp, times the recessive allele, and then multiply by 2, and that will get the frequency of our heterozygous dominant. So if we add the frequency of our homozygous dominant plus the frequency of our heterozygous dominant plus the frequency of our heterozygous recessive, that will equal 1. Because again, we're looking at frequency, and frequencies always have to add up to 1. So let's take this and put it into an example with our Hardy-Weinberg equilibrium. So we have a population of kangaroo rats, and we have a trait of interest. That trait of interest is the fur color. So we have 3 variations of fur color. We have the dark brown fur color, we have the spotted fur color, and we have the light brown fur color. Our dark brown fur is our dominant allele. So a dominant individual which has dark brown fur is going to have 2 capital Bs. Our heterozygous dominant individual, which actually turns out to be spotted, is going to have 1 capital B, so 1 dominant allele and 1 recessive allele. And then our light brown kangaroo rat, our recessive condition, is going to have 2 copies of the recessive allele. Ultimately, in our population of 1,000 kangaroo rats, so we're looking at 1,000 individuals for this population, we have 16 individuals of the population that are that have the homozygous dominant dark brown condition. We have 222 individuals that have the heterozygous dominant condition, or the spotted fur color. And then we have 762 individuals that have the homozygous recessive condition and have the light brown fur color. And what we can do is we can, based on those numbers, figure out the allele frequencies for our P, which is our dominant allele. That's the capital P, or capital B in this case, excuse me. And then we can figure out our Q, which is the recessive allele, or our lowercase b. So if we look down here where it says allele frequencies, for our P, our dominant allele, we are going to take 16 individuals, so that's all the individuals for the homozygous dominant condition, and multiply it by 2, because we have 2 copies of the homozygous, or we have 2 copies of the dominant allele. Then we're going to add those to 222, which is representative of the 222 individuals that have the capital B allele. And then we're going to divide that by 2,000. In this case, we normally think, oh, there's 1,000 individuals in the population. Why would we not divide by 1,000? Because each individual has 2 alleles, either a dominant, recessive, or a recessive and recessive. We have 2 alleles, so we're going to multiply 1,000 by 2 to represent 2,000 alleles. So we take the 2 times 16 and then add the 222 and divide by the 2,000 to get 0.127, which is representative of the frequency of our dominant allele, which is P. Then for our recessive allele, our Q, we're going to take 2 and multiply by 762 because there's 762 individuals that have the recessive genotype. We're then going to add it plus 222, which is representative of the recessive allele for our spotted kangaroo rat. There's 222 of those. We're then going to divide it by the total number of alleles, which is 2,000 overall, and thus get 0.873. And if we were to add these numbers together, we add 0.127 plus 0.873. That will equal 1 overall. So, taking this again one step further with our mating and producing offspring, we can then figure out the combinations or the frequencies of our genotypes for our offspring. So in this case, we're going to take an individual which is heterozygous mom and a heterozygous dad, and figure out all the possible genotype frequencies that we have. So we know the frequency of the dominant allele is 0.127, and the frequency of the recessive allele is 0.873. So using this sort of makeshift Punnett square, we're going to figure out the genotype frequencies. So P times P in this case is going to get our P squared, which is 0.016, which is representative of our homozygous dominant condition. So P squared is 0.16, which is representative by the P times the P. Second, we have our P, which is 0.127, multiplied by our Q, which is 0.873, to get 1 PQ, which is 0.111, but we do this 2 times. We do this in this top right square, and then we do this again in our bottom left square. So we have 0.111 for 1 PQ plus 0.111 for our second 1 PQ. For our heterozygous condition, however, we have to do 2 PQ, which is the sum of 0.111 plus 0.111. So ultimately 2 PQ will be 0.222. Lastly, in the bottom right corner, we have Q squared, which is representative of our homozygous recessive condition. So we multiply 0.873 by 0.873, and we get 0.762, which gets us the frequency for our genotype for our recessive homozygous condition. So ultimately, again, all 3 of these numbers for our homozygous dominant, our heterozygous dominant, and our homozygous recessive all add to 1, since they are frequencies. So the Hardy-Weinberg law is basically a very practical or a very theoretical application of how we determine the frequencies of not only the dominant and recessive allele, but also the frequencies for your homozygous dominant condition, heterozygous dominant, and homozygous recessive condition. Much like your Punnett square, though, your Hardy-Weinberg condition is very much theoretical. So there are a lot of conditions that have to be applied when we use Hardy-Weinberg, and they are these 5 that we see listed below. So the first one is that there's no natural selection, meaning that the environment in no way is shaping the direction of evolution. We don't have any form of mutation, so there's no spontaneous mutations. There's no mutations that are happening from external factors. We don't have any migration, so no individuals are leaving the population, nor are any individuals going into the population. There's no genetic drift, so we don't have any sort of founder effect. We don't have any sort of bottleneck effect ever in the environment. And lastly, there's random mating. So every individual is just mating with another individual. There's no form of choice or sexual selection or any factor like that in play. Ultimately, however, this is not very indicative of a real population. Of course, there's going to be mutations. Of course, individuals are going to leave and come into the population. There will be genetic drift. We do have natural disasters that occur from time to time. Random mating is not necessarily how individuals reproduce. Again, we have things like sexual selection and female choice that impact who mates with who. So Hardy-Weinberg equilibrium, much like your Punnett square, is a good model to help us understand the frequencies for an allele. However, it's not very practical on a real world level. So I just want to make that distinction very clear that we use Hardy-Weinberg as a tool. We don't necessarily say this is how the real world works. It's just a good a good theoretical application for looking at this concept. Okay, so I am going to stop it there, and I will pick up with part 2 shortly.